question. Perfect. So basically, what we have here is a three-dimensional image of fish, meaning that we were using fluorescent label props to localize the different structures that are chromosomes, but the chromosomes were identified in 3 dimensions. So basically, it's exactly the same. You have your sample on a slide. The sample could be a cell. Specifically, this cell, I believe it was in mitosis. And basically, the cell is stained with different props having different labels. And you're observing that each specific type of chromosome, which is each of these single colors is representing one chromosome, is labeled with that specific color. So basically what you're observing here is that the chromosomes are not linear, no? Don't have an X shape or a Y shape as we believe. Basically, they are like structures with 3 dimensions, but if you see, they look like, I don't know, like clusters, no? Like groups of different things with different colors. And if we move this is basically only a picture that is utilizing a specific and a specialized software, a very powerful software, that is going to allow to move the image in 3 dimensions. So basically, the only interesting part or novel part of this method is the use of a software, a specialized software, that is going to allow to capture the images and to convert them in 3-dimensional images, as you are observing here. So you can turn around the different structures, and you can see and visualize in different types of models. You can see this like sticks. Do you know what are sticks? Palitos, no? So you have a model with the sticks. You have a model with balls, tiny balls in here. And you have another model here where you are observing the distribution of that chromosome in the space. So this looks very different from the pictures that we traditionally see, no? Do you have any questions with this? The only difference is the microscope that is utilized to capture the images. The name of the microscope is confocal microscope. And then the second difference is the software. The software that is utilized to record the image and to manipulate the image is different from the other examples that we have seen. What? Okay. The next one is called halo fish. For this one, once again, we're observing the sample. The sample is a cell. The sample is highlighting the nucleus. This is the nucleus. So basically, the cells are attached into a slide, once again, a glass slide, and then they are treated with a specific reactive that is going to allow the, how can I say? It's going to allow the release of the chromatin, because the reagent is going to make holes in the nuclear envelope. And basically what you're observing here in purple are the chromosomes that are liquid outside the different nucleus pores. And you're observing that we are having something similar to a halo. That is why it's called halo fish. And then, take a look. This is already fixed inside the nucleus. The nucleus is not totally destroyed. It's partially destroyed only in the nuclear envelope. And that's why the chromosomes can leave a little bit the nucleus. But they are not abandoning the nucleus because they are still attached to it. So basically, these portions that are outside the nucleus are the ones that are treated with the different props. This is a prop. The prop could be fluorescent because we are talking about fish. It's going to have a fluorophore attached to the prop. And the probe is going to be left to hybridize. But it's going to be hybridized only with the DNA that is outside the nucleus, because cannot reach the inside of the nucleus. And outside here, we are going to observe the hybridization, and the hybridization can later be observed by microscopy or can be observed by electrophoresis. Okay? In microscopy, we are going to observe, the picture is very tiny, but the nucleus is going to be here, and the hybridization is going to be observed all around the nucleus in a halo. That's why the name is halo fish. Okay? Yeah. Questions with this? Okay. The last one is this one. This one is utilized to identify changes in the chromosomes, changes that are affecting or causing a disease. What type of changes? We have several types of changes. Translocation. What is a translocation? The change of a fragment of a chromosome into another chromosome or another region of the same chromosome. I'm going to repeat again. Translocation is the change or the movement of a specific fragment of DNA into another chromosome or into another part of the same chromosome. That is a translocation. So we're observing here that we can detect or we can design proofs directed to regions on the chromosomes that usually translocate. So basically, if the probe doesn't find that specific region or doesn't hybridize against that specific regions, that means that the region was translocated to another place. So you can detect translocations by splitting the different regions, or you can identify translocation by doing fusion probes. What is that? In the case of, probably it's going to be better if I draw. Okay. So you have two options in order to do this analysis of translocation. If I have this portion of this gene translocating, what is going to happen with this portion of the gene? This portion of the gene is going to change to another chromosome, like here, for example. Okay? This is a translocation. However, if I want to detect here this region, I will design a probe against that region, right? But what is going to happen if this portion is already translocated to another position? The probe is not going to find it, and the probe is not going to do a hybridization. Yes or no? Yeah. However, I can do the other or the opposite part. I can look for the region in another chromosome. Chromosome 2, chromosome 1. If the same probe is incubated with another chromosome and is showing a signal in another chromosome, that means that this region was translocated or moved into another chromosome. So in this way, I can do two types of analysis. I can look for the absence of fluorescent signal, or I can look for the presence of fluorescent signal in another chromosome. And I can determine a translocation present in there. Those are the two examples that we have here. We can also identify something that is called amplification of chromosomes. What is that? In some cases, some specific part of the chromosomes, genes, specific genes, the ones highlighted here in red, are copied several times and not only one time. This is a normal chromosome. Only one copy of the red gene and one copy of the green gene is present. However, due to an abnormal activity of the DNA polymerase, one gene can be copied several times, and we can have several copies of this gene. This is abnormal. This can cause a disease. This can be detected because if I do a probe, directed against this red, here is the probe, directed against the red gene. If I have a normal, the signal is going to come from only one probe hybridizing this gene. However, in the abnormal gene, I'm going to have several of them. And then I'm going to have several probes attaching here. Then the fluorescence is going to be higher than this. Yes or no? So by detecting differences in the intensity of the probes, in the intensity of the fluorescence, I can detect this type of abnormalities. The last one, the following one. What we're observing here is a deletion. Remember, a deletion is the lacking of a region in certain genes or certain parts of the chromosome. How this happened? This happened because there are endonucleases that cut parts of the chromosome. So basically you're observing here that the same gene in red is present in both chromosomes. What happened here is that only one of the chromosomes have the gene and the other has already lost that specific gene. So basically that means that the probe is going to hybridize here. The probe directed to this gene, the red one, is going to hybridize here, but it's not to be hybridizing here. And then I'm going to notice a difference in the intensity if I compare a normal chromosome against a deleted chromosome. Okay? And the last one, polyploidy. Polyploidy, once again, if I have, instead of having two chromosomes, there was a mistake and the polymerase made another extra chromosome. We have three. We have a trisomy here. The intensity of the fluorescence is going to change too, because normally I only have two chromosomes. I will see a certain level of fluorescence. But if I have another chromosome with the same regions, the probe is going to hybridize to that chromosome, and the fluorescence is going to increase. So that change in the intensity of the fluorescence can be detected. So all of these modifications that I just highlighted here can be detected with fluorescence or with FISH. And the name of this is a split signal fluorescence. So I'm talking about intensities of fluorescence here. Do you have any question? No. No? Okay, that's it. So the evaluation is going to be until here, since the beginning of Unit 2, okay? And it's going to be on Monday. That's it.